The next variant of the PCR is quantitative PCR or qPCR. Quantitative PCR or qPCR is used to measure the quantity of a target sequence in real time. It quantitatively measures starting amounts of the DNA or initial quantity of the DNA, cDNA or RNA in the sample. Quantitative PCR is commonly used to determine whether a DNA sequence is present in the sample and the sample of the number of copies of these DNA in the sample that can be determined using whether it is present or not, that can be determined. Also, the quantity of the DNA initially present in the sample that can also be determined using quantitative PCR. Quantitative PCR has a very high degree of precision compared to the normal PCR. And these methods use fluorescent dyes such as cybergreen, evergreen or fluorophore containing DNA probes such as TACMAN to measure the amount of the amplified product in real time. It is also sometimes abbreviated to RT-PCR or real-time PCR. But this abbreviation should be used only for reverse transcription PCR, not real-time PCR. qPCR is the appropriate abbreviation for a quantitative PCR or the real-time PCR. Whereas the RT-PCR, that term or that abbreviation is used for a reverse transcription PCR. So you have to keep it in mind. So the RT corresponds to reverse transcription, not real-time. Real-time PCR is known as a qPCR. So this is an illustration of qPCR in which here we have the template DNA which will be bound with a probe, a fluorescent probe with a reporter and a quencher. And this is the polymerase, and during the amplification the polymerase will move through and it will remove the reporter and quencher. It will be cleaved. This and the removal of these specific reporter fluorescent probe from the DNA by cleaving activates the fluorescent component of the fluorophore, and that fluorescence emitted can be detected in real time. So for every step of amplification of this template, you will have fluorophores released from the sample, and the fluorescence can be quantitatively determined, which corresponds to the initial quantity of the DNA, and the subsequent amplification of the template DNA can be quantitatively measured using by measuring the intensity of the fluorescence released by the fluorophore. So this is a fluorescent emission, and that is measured in the qPCR system. I will show you one video.
In contrast to traditional PCR, where the presence or absence of a DNA sequence is analyzed upon reaction completion, quantitative PCR, or qPCR, monitors DNA amplification as the reaction progresses. In qPCR assays, reaction progression is monitored by an increase in fluorescent signal. The fluorophore can be a double-stranded DNA binding dye, most commonly cybergreen 1, or covalently attached to a target-specific probe, as in TACMAN assays. In cyber assays, the dye fluoresces when bound to double-stranded DNA, but displays weak fluorescence in the presence of single-stranded DNA. This results in an increased fluorescent signal as the reaction progresses, and double-stranded DNA is formed. The most common probe-based assay is a 5' nuclease assay, known as TACMAN. In TACMAN assays, the 5' flapped endonuclease domain of Tac DNA polymerase cleaves a target-specific probe, separating a fluorophore on the 5' end from a quencher on the 3' end, resulting in an amplification-dependent increase in fluorescence. A thermocycler, coupled with a fluorimeter, modulates the reaction temperature during DNA amplification, while also monitoring the fluorescent signal at each PCR cycle, generating a real-time qPCR curve. There are 4 distinct phases within the amplification curve, lag, exponential, linear, and plateau. During the lag phase, amplification occurs, but the signal is not strong enough to be detected above background. The exponential phase is used for quantification because it produces a fluorescent signal measurable over background and contains an abundance of reaction components, permitting a detectable doubling event at every PCR cycle. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle, or CQ value, can be determined. CQ values can be used to evaluate relative target abundance between two or more samples, or to calculate absolute target quantities in reference to an appropriate standard curve. qPCR can also be modified to detect and quantify RNA. Interpretation of qPCR data requires careful experimental design and analysis.
Okay. So that's about the qPCR. Now the next one is RT-PCR, or the reverse transcription PCR. And now you all know about the concept of the RT-PCR, especially in the year of COVID-19. The reverse transcription PCR or RT-PCR is for amplifying DNA from RNA. So for an RNA virus, especially for the detection of the RNA virus, the reverse transcription PCR or RT-PCR is the best tool for confirmation. The reverse transcriptase, that enzyme reverse transcribes the RNA into a cDNA, which is then amplified by the PCR. So it basically involves two steps. One, production of the cDNA from the RNA by reverse transcription. Then subsequent amplification of the cDNA or DNA, multiple DNA copies can be produced to produce sufficient quantity of the DNA for detection. So, sufficient templates should be there for reaching the detectable limits, and that means it needs initial reverse transcription of the RNA to cDNA, and subsequently the cDNA will be again amplified using the normal PCR, so that a large quantities of the template DNA will be produced, which can be detected using the detection system. So the RT-PCR is widely used in expression profiling to determine the expression of a gene or to identify the sequence of an RNA transcript, including the transcription start and termination sites, etc. If the genomic DNA sequence of a gene is known, then RT-PCR can be used to map the location of exons and introns in the gene. And then the 5' end of a gene, which corresponding to the transcription start site, is typically identified by RACE PCR, which is known as rapid amplification of cDNAs, which is another variant of the PCR. So this RT-PCR is having various roles: gene expression analysis and viral RNA detection, then identification of the 5' ends of the gene. So there are a lot of variants of the RT-PCR, application of the RT-PCR nowadays available. So this is the concept where we have the RNA that needs to be detected using a specific primer. The initial step is the reverse transcription stage, in which the cDNA copy is produced, and then using the PCR with from the cDNA, multiple copies of the DNA can be produced by PCR amplification. So this is the normal amplification of the PCR, and so there should be a reverse transcription phase. There should be a PCR amplification phase, second stage. So these two-stage process will result in sufficiently enough quantities for the DNA in the sample for easier detection. So this is the first step. Once again, we have various reactants that is available, like DNA polymerase, then reverse transcriptase, both the enzymes is required, then other nucleotides or DNTPs, then DNA template, water or buffer, etc. All these things will be mixed in the same reaction mixture, and the first step, that is real-time PCR and the PCR, both were occurring simultaneously in the same reaction mixture, So this is a single step, one step RT-PCR in which everything is mixed in the same vial and simultaneously. And after completing the first step of reverse transcription, the DNA polymerase will do the second step, and finally you will get the amplified DNA in the same reaction vial. And in two-step RT-PCR, we have initially the first step that is conducted that is done by the reverse transcriptase enzyme, which will produce these cDNA copy. And in the second stage, you will add the DNA polymerase enzyme like TAC or something, which is required for DNA amplification using this DNA or cDNA copy which is present in the vial after the initial amplification. So this DNA polymerase which is added will do the second step, and finally you will have the product that is the PCR product that is formed. So you can do this reverse transcription PCR in single step with all the reactants mixed together in single step, or you can do it in the two step with the first RT reverse transcription step, and the second one is the normal PCR step. So both the ways you can do the reverse transcription PCR or RT-PCR. Again, I will show you one video. You can see that one.
Reverse transcription is the process of making complementary DNA, cDNA, from single-stranded RNA. The experiment requires RNA, a reverse transcriptase enzyme, a primer, and dNTPs. The RT experiment is also called first-strand cDNA synthesis. The steps are as follows. RNA is first incubated with a primer at 70 degrees centigrade to denature RNA's secondary structure. This is chilled quickly on ice to let the primer anneal to the RNA. Reverse transcriptase, dNTPs and buffer are added to the reaction. RT reaction is extended at 37 degrees centigrade or higher for 1 hour to allow transcription to occur. RT reaction is heated at 70 degrees C to inactivate the enzyme. The end result is cDNA. There are many reverse transcription kits available that all address different concerns. We are going to cover a few of your choices here. Enzyme choice. For reverse transcriptase, we have several options. A good basic enzyme is MMLV. This enzyme will get the job done, but there are limitations. You can only perform your RT up to 37 degrees centigrade, which can be a problem if you have a GC-rich template or lots of secondary structures to deal with. A better reverse transcriptase is superscript 3. Superscript 3 is a highly thermostable proprietary mutant of MMLV enzyme with reduced RNase H activity. This engineered reverse transcriptase allows the RT reaction to go a higher temperature of 50 degrees C. This means that you get more yield, longer products and greater representation of your input RNA. Can superscript 3 be used for real-time PCR? For real-time PCR application, you may have high and low expressing transcripts in the same sample, and you only want to quantify the target of choice. Therefore, you want to have a high sensitivity and specificity as demonstrated with lower CT values and single peak in melting curve analysis. Superscript Vilo contains superscript 3, the same thermostable RT that we've talked about before, but with an optimized buffer mix and helper proteins. It's specifically designed for generating cDNA for real-time applications, thus resulting in high yields and unprecedented linearity across a broad range of sample outputs. The common choice of primer is oligo-DT. It binds to the 3' poly A tail of eukaryotic mRNA transcripts. The limitation with this choice is that you can get a 3' bias of your sample due to where the primer binds. In addition, oligo-DT is not compatible with non-eukaryotic or degraded samples due to the lack of a poly A tail, and hence the oligo-DT primer cannot bind. An alternative is random primers, which are randomized hexamers or non-amers. These primers will bind all along the RNA transcript and so help to overcome issues with secondary structure and give a good representation of the population. Random primers usually result in higher cDNA yields. Some researchers choose to use a blend of both random primers and oligo-DT to get the benefits of both options. The final option for primer choice is gene-specific primers. These will only make cDNA for the one specific target. Gene-specific primers are required for one-step real-time PCR, RT-PCR reactions. The cDNA yield will be lower with the gene-specific primers since you are only targeting one transcript.
There are two primary approaches to reverse transcription for PCR, the one-step approach and the two-step approach. In two-step RT-PCR, the first step involves the reverse transcription, or RT, of RNA by a reverse transcriptase. This reaction generates the first strand complementary DNA, or cDNA. cDNA is then transferred to a separate reaction tube for the second step of this process. During the second step, the second strand cDNA is generated and amplified by a DNA polymerase. So, in two-step RT-PCR, reverse transcription and PCR occur in separate reaction tubes. This approach is used when there's a need to analyze multiple targets from the same RNA sample in separate reactions or to store synthesized cDNA for later use. In the other approach, known as one-step RT-PCR, the reverse transcriptase and DNA polymerase are pre-mixed, which allows the RT and PCR steps to be performed in the same reaction tube. Not only is the one-step RT-PCR approach faster, it also requires less pipetting and minimizes possible contamination, thereby improving data reproducibility. It also enables easier processing of large numbers of samples for high-throughput applications. The Invitrogen Superscript 4 one-step RT-PCR system combines the Superscript 4 reverse transcriptase with the high-fidelity platinum superfied DNA polymerase to allow highly efficient one-step RT-PCR. This system offers high sensitivity. It allows one-step RT-PCR of a broad range of target lengths, and it works even in the presence of common reaction inhibitors. The system also supports a multiplex one-step RT-PCR approach and is compatible with high-throughput applications. Come one step closer to superior RT-PCR results with the Superscript 4 one-step RT-PCR system.
Okay. So now we will start with the next PCR, that is touchdown PCR. Touchdown PCR, or it is known as the step-down PCR, is a variant of the PCR that aims to reduce non-specific background by gradually lowering the annealing temperature as PCR cycle progress. So it actually starts with a high annealing temperature initially, and during the subsequent cycles, it continuously brings down the annealing temperature, and that is why it is known as touchdown PCR. The annealing temperature at the initial cycle is usually a few degrees, 3 to 5 degrees above the TM of the primers used, while at the later cycles, it is a few degrees, 3 to 5 degrees below the primer melting temperature. When we discussed the primer design concept, we said that the primer annealing, the annealing temperature should be about 3 to 5 degrees Celsius above the melting temperature of the primer. But that is a normal requirement for the PCR. But in touchdown PCR, we start with a high temperature for annealing than the primer melting temperature. But then we bring it down during the subsequent cycles. The annealing temperature is brought down below the primer melting temperature, about 3 to 5 degrees Celsius below the primer melting temperature. The higher the temperature is given, that gives the greater specificity for primer binding, and the lower temperatures permit more efficient amplification, but non-specific bind products may be formed at lower temperatures. So the touchdown PCR initially ensures that at higher annealing temperature, more specific amplification is ensured, because you are setting a high temperature initially, but after a few cycles, since there is a sufficient quantity of the templates already produced, you can bring down the annealing temperature, and non-specific binding will not be an issue with more templates available after the initial cycles of amplification. So this is the representation in which, for example, initially it is denatured at 95 degrees Celsius for 5 minutes. Then you can use 58 degrees Celsius for 30 seconds. Then cycled 20 cycles. After 20 cycles, you will bring down the annealing temperature from 58 to 48 degrees Celsius. 48 is actually a lower temperature, which even facilitates non-specific binding, but reaction will be much faster, and sufficient binding and amplification can be ensured at this stage. But if you are initially using this 48, the non-specific binding products will be much more. But in this case, the first cycle, 20 cycles, were set at a high temperature of annealing, so that the non-specific binding is not occurring at that stage. But after 20 cycles, you have sufficiently larger quantities of the template DNA available, and non-specific binding will not occur after that one. So after some cycle, 20 cycles, you will bring down the temperature, annealing temperature, to 48, and again 10 cycles will be conducted, so that increased amplification can occur at that time. Then finally you will close the cycle with a one cycle at 74 degrees Celsius. So this is a 4 degrees Celsius storage and end of the cycle. So this is the procedure for a touchdown PCR, in which you will gradually bring down the annealing temperature and increase the efficiency of the amplification. The initial high temperature set, that is used for increasing the specificity of the amplification. So the next one is asymmetric PCR. Asymmetric PCR, preferentially it amplifies one DNA strand in a double-stranded DNA template. Normally, a normal PCR amplifies both the strands with the two sets of primers. Two primers or a primer pair. So both the strands will be equally amplified. But if you are targeting a single strand amplification, not the amplification of both the strands of the DNA, then asymmetric PCR is the choice. It is used in sequencing, where actually for DNA sequencing you need only sufficiently larger quantities of a single strand of the DNA. So if you know the sequence of one strand, the other complementary strand can be easily predicted. So you don't need to amplify and sequence both the strands of the DNA. So in the sequencing process, you actually require one strand of the DNA amplified in sufficiently larger quantities, for which asymmetric PCR is a choice. So PCR is carried out as usual, but with a great excess of primers for one strand targeted for amplification. So you will control the quantity of the primers in such a way that one primer or the target strand primer can be used in excess, whereas the other one will be a limiting factor. So instead of using equal proportion or equal quantity of both forward and reverse primers, one primer will be used in larger quantity or greater quantities or in excess, and while the other one will be a limiting one. Because of the slow or arithmetic amplification, later in the reaction, after the limiting primer has been used up, extra cycles of PCR is required. Which means instead of it's not a chain reaction. Like a logarithmic amplification is not possible since one of the primers is a limiting factor, so usually it amplifies in an arithmetic manner, like for example, instead of doubling the strands, one will become 2, 2 will become 4, 4, 8, and subsequently only it will it will increase in an arithmetic fashion, not in a logarithmic fashion. And a recent modification of this process known as a linear after the exponential PCR or late PCR is used. It uses a limiting primer with higher melting temperatures Tm than the excess primer to maintain the reaction efficiency, as the limiting primer concentration decreases mid reaction. After some time, after the limiting primer is consumed, or all of the limiting primers were consumed, then the reaction almost stops. But in that case, you require some other modification like late PCR. Anyway, instead of using a logarithmic scale of amplification, this arithmetic PCR, asymmetric PCR is using arithmetic amplification of the template based on a single primer. So you can see this one. So one primer is an abundant one, and the second one is a limiting primer. So one of the strand, or the non-targeted DNA strand, this is a amplified using the abundant primer. So based on the number of copies of the primer available, larger or increased quantities of the of a single strand is produced, whereas the limiting primer will amplify only the targeted strand. A few copies of the targeted strand is produced by the limiting primer. So this is how the asymmetric PCR amplification works. The next one is allele specific PCR. In allele specific PCR, actually it is a diagnostic or cloning technique based on single nucleotide variations. It is SNV, SNP, it's not SNP, not be confused with SNP or single nucleotide, single base, single nucleotide polymorphism. This is a single nucleotide variation, SNV. And this actually means a single base differences in one patient or one organism. It requires prior knowledge of the DNA sequence, including differences between alleles, and use a primer whose 3' N encompasses the single nucleotide variation. So you design a specific primer which is targeting the gene with the variation or gene with a single nucleotide difference. So the wild type will have a normally used nucleotide, but the patient or the diseased gene that you are looking for may have a single nucleotide variation. So you design a primer which will target that single nucleotide variation if present. That will be targeted and it is amplified. So the PCR amplification under stringent conditions is much less efficient in the presence of a mismatch between the template and the primer. So successful amplification with an SNP, specific primer signals the presence of a specific SNP in the nucleus. So that means if you are designing a primer which is targeting that single nucleotide variation, and if you are getting an amplification, that means in the target DNA that mutation or that variation is present. So if it is not, that variation is not present. So you are designing the primer which is specific for that corresponding variation. So this is another.
With the new Life Technology developed qPCR technology, somatic mutations now have nowhere to hide. Pacman mutation detection assays use this qPCR technology, which refers to competitive allele-specific Pacman PCR, specifically designed to detect somatic mutations that occur in cancer cells. Let's see how it works. Let's take an example of a tumour sample in which only 0.1% of the DNA contains a somatic mutation and the other 99.9% is normal wild-type DNA. A mutant allele assay based on qPCR technology contains 4 components. The allele-specific or AS primer is designed to be specific for a mutant allele in a target gene. A locus-specific primer is designed to be specific to the target gene. This will bind to mutant and wild-type allele containing DNA. A locus-specific TACMAN probe is designed to bind to the target gene between the primers. This will also bind to both mutant and wild-type allele containing DNA. This probe will generate a fluorescent signal during the PCR reaction to monitor the target amplification. An allele-specific MGB blocker oligonucleotide is designed to bind specifically to the wild-type allele. The role of this blocker is to compete with the mutant allele-specific primer and inhibit its binding to the wild-type allele. This blocker suppresses non-specific amplification of the wild-type allele and improves the specificity of the reaction. Only the mutant allele is amplified and generates a fluorescent signal. By increasing the specificity and inhibiting the amplification of the wild-type allele, this competitive allele-specific TACMAN PCR provides a very sensitive and specific method to detect as low as one mutant allele in a presence of 1,000 wild-type alleles. Find the TACMAN mutation detection assays you need at lifetechnologies.com/castPCR.
Gibson assembly, developed by Dr. Daniel Gibson and his colleagues at the J. Craig Venter Institute, is an effective method for the assembly of multiple DNA fragments. This is accomplished in a single-tube isothermal reaction with Gibson assembly master mix. The method utilizes adjacent DNA fragments with complementary ends, which can be added, for example, by PCR. Then, the overlapping fragments are added to the Gibson assembly master mix and incubated for 1 hour at 50 degrees Celsius. During the 1-hour incubation, the master mix's 3 enzyme activities set to work on the fragments. First, a 5' to 3' exonuclease activity creates single-stranded 3' overhangs. These complementary sequences then anneal, creating the double-stranded DNA of interest. DNA polymerase then extends the 3' ends, filling in the gaps, and DNA ligase seals the remaining nicks. The end result is a fully sealed double-stranded DNA molecule that can serve as template for PCR, RCA, or a variety of other molecular biology applications, including direct transformation. Gibson assembly has been successfully used to reliably join up to 6 DNA fragments into a single molecule. This remarkably straightforward and powerful technique makes quick work of large multi-fragment assemblies, but it is also useful for more routine applications, such as cloning. The Gibson assembly cloning kit, which includes both Gibson assembly master mix and NEB5 alpha competent cells, has been optimized for efficient assembly and cloning of multiple fragments into any vector in just under 2 hours.
Another one is a digital PCR or dPCR. And digital PCR is used to measure the quantity of a target DNA sequence in a DNA sample. The DNA sample is highly diluted, so that after running many PCRs in parallel, some of them do not receive a single molecule of the target DNA. So usually the DNA sample is highly diluted, and maybe some of the reactant vials or wells will not receive a single template DNA for amplification. The targeted DNA concentration is calculated using the proportion of the negative outcomes, and hence the name digital PCR. So it's calculated. The target DNA concentration is actually calculated based on the outcomes, and that means that's why it is known as a digital PCR. It's a calculated outcome. So you will see another illustration here.
Digital PCR is a next-generation approach for specific detection and quantification of nucleic acid sequences. PCR has the ability to generate detectable quantities of specific DNA sequence from a single molecule through the exponential amplification of sequences. This is done through the use of primers and a thermostable DNA polymerase. By monitoring this amplification in real time, quantities can be measured with great accuracy. Since this is compared to an external reference, measurements are relative quantifications. Digital PCR builds on these same principles of DNA quantification, enabling an absolute value with much higher precision compared to standard real-time PCR, and without the need for any reference. If you are already doing real-time PCR, moving toward digital PCR is straightforward. Your sample preparation and TACMAN assay remain the same. Just swap out your real-time instrument with a standard thermal cycler and digital PCR instrument. Digital PCR starts with the partitioning of your reaction mix into hundreds to thousands of independent PCR replicates. Some partitions receive the nucleic acid sequence of interest and others do not. Partitions with a target molecule will amplify via PCR to a detectable level. These are categorized as positive reactions. Partitions without the target molecule will read as negative reactions. In the end, the number of positive reactions is directly proportional to the total number of molecules present in a sample, enabling an absolute count to be established. To understand the absolute count, it's necessary to understand that some reaction wells will receive more than one target molecule during partitioning. Uncorrected, this can lead to undercounting, as only positive reactions are counted. This mathematical issue may seem daunting, but fortunately a digital PCR instrument, such as the Quant Studio 3D from Life Technologies, figures this out automatically with the use of standard statistics. Following identification of positive and negative wells, a Poisson model is used to calculate the probability of a given reaction receiving 0, 1, 2, 3, or more copies. This correction factor enables all molecules in the starting sample to be accounted for, yielding absolute quantification. Within absolute quantification are performance attributes that enable applications to go beyond the limits of real-time PCR. Digital PCR enables increased sensitivity and specificity of target detection as the partitioning of samples into individual reactions reduces competition of background DNA. Volume sampled also affects sensitivity. For example, if an inadequate volume is sampled, there isn't enough sensitivity to detect a rare target. Increasing the volume sampled naturally increases sensitivity. Precision is another performance attribute of digital PCR. This is driven by the number of replicates or partitions that are run. Increasing replicates increases the statistical significance of your answer, thereby giving more confidence that the value determined represents the actual target quantity in the sample, and makes it possible to directly compare results across experiments and labs. And with these qualities, digital PCR is ideal for applications such as rare mutation detection, pathogen detection and quantification, genetically modified organism detection, accurately quantifying NGS libraries and validating sequencing results, and developing a reference for your real-time experiment when it doesn't exist, just to name a few. While real-time PCR still maintains its advantages of enabling higher throughput and a wide dynamic range for your assays, you'll find that digital PCR is a great complement to real-time PCR. This is evident in cases of a very late CT in real-time experiments, where the precision in the measurement is not adequate to make a determination. The added sensitivity and precision of digital PCR can rescue your results, eliminating the need to redesign experiments. Digital PCR application possibilities are extensive, and it's never been easier to put the latest technologies right at your fingertips. What will you do with digital PCR?
So the next topic is about ligase chain reaction. And ligase chain reaction amplification of genomic DNA is done something like this. We can see that there is a primer, two primers, primer 1 and primer 2, that is against two targets. So ligase chain reaction is not an amplification, it's a ligation process of primers. So the target DNA sequence is amplified by joining of the primers. So the target DNA is not amplified, it is actually a pre-synthesized primer, two primers, and this will be ligated and joined so that this is not a amplified template or amplified copy of the DNA. That specific primer which will bind to the target will be ligated together to form the new template or new copy. And this cycle continues until the ligation of the two primers and construction of the next template, which results in the construction of the next template will be done as a chain reaction. So that is why it is known as ligase chain reaction. Here there is no polymerase, no amplification, only ligation process is there, and the joining of the two primers against the targets will result in the production of the new copy of the template, and that cycle continues in the just like a chain reaction. So this is about ligase chain reaction. So we have the target DNA. For the identification of the target DNA, you design two specific primers against the target DNA. Once it is joined and ligated, you have a new template that is available. This will act as the template for the next joining of the primers. So this is another representation. Here we have the template. These are the probes of the primers that can bind here. So both the strands will have the concerned primers designed that will be joined here, and it will be ligated to form the new strand. And the second strand will also be joined with the concerned primers, and that will produce the second strand. Now again, denaturation and second cycle starts, where the new primers will bind or the probes will bind, and they will be ligated together, and this cycle continues, which will act just like a chain reaction. So the next type of PCR that we want to discuss is overlap extension PCR. In overlap extension PCR, here we have this is a case where you can join together two strands. Like here we have one primer that is used with an extension or an added extension on one end. So one of the primers will have a tail sequence. That's an extension additional sequence, and the other strand is amplified with another extension. So these two extended regions will be complementary, and finally, initial stage of amplification, you will have the after the first cycle of amplification, the second strand is synthesized against these newly added region also. So now you have both the strands amplified with the added regions or newly added regions. Now they can be joined or mixed together and ligated, so that mixed together and hybridized. So the strands will overlap in these new overlapping regions. Newly added overlapping regions will be used for joining of these two strands. And now finally it is the strand is synthesized, and that overlap extension PCR is now used for joining together of two DNA strands. And there is another role. It is used for side-directed mutagenesis. This type of overlap extension PCR is used for a side-directed mutagenesis. This is another illustration in which we are using two pairs of primers, primer 1, primer 2, and primer 3 and primer 4. Four primers were used for introduction of such side-directed mutagenesis in overlap extension PCR. In the PCR step 1, you will use say primer 1 and P1 and P4 is used here. Then P3 and P2 were used in this reaction. So the products will be something like this. P4 will amplify this product, P1 will amplify this product. Then the other one, P2 will amplify the longer product, and in PCR2, P3 will amplify the shorter fragment. Then after the synthesis and separation of the strands, you can join them together, and you have multiple PCR cycles where the fragments will be amplified, then they can be denatured and annealed together, and that can be joined so that the entire strand is joined together to construct the mutated gene. So where you are incorporating the mutation? In the primer 4 and primer 3, if you are changing some of the bases in the primer 4 and primer 3, that change will be subsequently incorporated in the amplification stages, and that mutation will be incorporated in the position of primer 3 and 4. So if you want to introduce a site-directed mutagenesis, you design a primer 3 and primer 4 with the desired mutation. Then after the amplification, two-step amplification and subsequent joining or overlap extension of this PCR, your desired mutation, single base mutation or site-directed mutation, is introduced in the mutated gene. So such type of site-directed mutagenesis is possible using overlap extension PCR with such modifications in the primers introduced in the second set of the primers. So this is about overlap extension PCR. Now the next one is rolling circle amplification or RCA, in which we have the ligation template, which is here and represented here. This is a circular template DNA. This is a ligation template. So the circular DNA is joined together after annealing, and the ligation template is presented here. And once it is ligated, now you have the circular DNA with a ligation template, and that can be purified. And this purified circular DNA can be added with a primer, and that primer will produce a longer DNA copy using rolling circle amplification. So here we have different detection systems for such amplification products. In the first one is a dye-labeled DNDP labeled detection system. The second one is a probe-based system, and the third one is a molecular beacon-based system, where we have a fluorophore and a quencher that is used just like that of the qPCR. We have a molecular beacon-based system for detection of the amplification. So these are three different possibilities of detection, like dye labeling, DNA probes, radio-labeled probes or something, and molecular beacons. So this can be attached with the newly synthesized DNA for their detection. So rolling circle amplification initially circularizes the template or the target, then add the primer and amplify using rolling circle replication. So multiple copies of a concatemer of the entire target DNA can be synthesized by rolling circle replication. Here again we can see there is a various stages, like the previous one is explained here, and we have the rolling circle replication here, which produces this is the DNA copy, and the same thing can be created using RNA copy also. So here we have three different cases, like the first one, E is exponential DNA amplification by hyperbranched RCA. So this is a multiple polymerases were binding, DNA polymerases were binding to the newly synthesized DNA. After the rolling circle replication, we have a long strand of the DNA which is created here, which will be bound with a DNA polymerase, and multiple templates will be produced or copied from this available template. So this results in large scale amplification of the target DNA. So this is the target DNA which is circularized. Initial DNA copy is produced using the rolling circle replication. Then the DNA polymerase will bind to that template and amplify the longer stretch of the DNA using primer 1, primer 2, or primer 3, primer 4, etc. So all the primers will simultaneously amplify, producing sufficiently larger quantities of the product. So in the case F, where exponential DNA amplification generating multiple circles of the product is produced. For example, here is the initial copy of the rolling circle replication. Then you can complementary to restriction site is there. Restriction enzyme digestion is done. So it will create multiple copies, and each copy can be circularized. So initially the DNA is actually broken into fragments, then circularized into different circular DNAs. again the rolling circle replication model is done. So that is another method. So in this case, we are using a nicking enzyme for breaking down the initial RCA amplification product, and that will be circularized. Then using a template that can be bound over a template or a circular template. Then DNTPs were used for amplifying these templates. So this type of exponential DNA amplification by primer generation using nicking enzymes. That is another possibility. So this is another method of RCA amplification. And in all these methods, the initial template is amplified in sufficiently larger quantities. So this is another illustration in which this is a dye labeling of target DNA. This is a molecular probe, like a radio labeled probe with which is labeled with a primer. That can be another type of DNA labeling. This is a molecular beacon with a fluorophore and a quencher that can be attached, and the removal of this fluorophore probe will give you a fluorescent signal that will enable EC detection, just like that of the qPCR. So this is a different examples of detecting the amplification products.